true, okay? But generally speaking, it is. All right, so we talked about all this stuff. Oh, and one other thing with the portal of entry that we forgot to mention, the inoculum. So what is the inoculum? So in lab, you're inoculating, right? And inoculating is taking organism and putting it into another area or tube. So the inoculum is the amount of organism that you are transferring, okay? And when we're talking about disease, that means the amount of organism that gains entry into the body, okay? And this is known as the ID. And this is important because it tells you how serious the infection may become. And then in case you forgot about toxins, or thought we never talked about them, here's the slide that shows them. Okay, now the rest of this chapter is a lot of just definitions and boring stuff. Okay, so we're just going to fly through it. So, how do infections establish themselves? Well, they stay in one area. This is a localized infection. They get into the bloodstream. This is a systemic infection. They start in one area and then move to another area. This is known as a focal infection. If you have multiple organisms in one place, this is known as a mixed infection. And then if you are infected with a primary pathogen, or one that causes disease naturally, and this preoccupies your immune system and then get infected with another organism, this is known as a secondary pathogen. And this shows what happens to an individual during the infection we talked about previously. Okay? So when you first get infected with your organism, you go through a period with little symptoms. This is known as the incubation period. Okay? Remember, this is where there's not enough organism for your immune system to respond yet, so you don't feel sick, but the organism is there and it's starting to grow. Okay? Then it will start to grow and your immune system responds, but in a very non-specific way. This is where you develop the prodromal stage of the infection. You develop symptoms, but they're much more mild and nondescript. And so this is where the organism is just starting to gain steam. Then the organism starts to grow and divide very quickly. The immune system is trying to play catch up. And at this period, or at this place in time, you're going through the period of invasion. And this is where you will develop the most intense signs and symptoms. And these will also be the signs and symptoms associated with that particular disease. Hopefully your immune system kicks in. This is where you reach the height of infection. The organism is kind of kept at bay. And then eventually, if the immune system kicks in fully, you should clear the organism and your symptoms will decrease rapidly. This is known as the convalescent period. And the goal of this is to reach full recovery, which is where all organism is removed from the body and you are back to normal health. So what is a sign? Can you remember? Objective measurement that a clinician can do, right? So temperature, vitals, things like that. What about a symptom? So subjective description, usually from your patient, right? So pain, malaise, sleepiness, things that can't actually be measured well. Then what about a syndrome?
You can actually identify what the person has.
Right, so this is all the signs and symptoms associated with a particular disease. Okay? And you can use that for differential diagnoses to come up with what the person has. You got this sign, this sign, this sign, and this symptom. That means you have such and such disease. Go ahead. What is latency? A dormant state. You can read. Good. What does that mean?
It means the bacteria is hiding between someone, waiting for.
Right. They get in and then they don't do anything. They don't cause disease. They just hang out. Right? And then 10, 20, 30 years later, they wake up and then you get sick again. What about a sequelae? So sequelae are long-term destruction in tissues that cannot repair themselves. And so even after you clear the pathogen, you still will have problems with that tissue. Okay? Sure.
People that have shingles and they have pain for years.
Phantom pain?
Yes.
Yeah, yeah. The only problem with phantom pain is we don't really know why it happens. So it gets kind of hairy. Because normally with sequelae, it's damage that can be detected, right? And so there's probably nerve damage, we just can't detect it. So it's kind of in a gray area. But most of these sequelae, at least with the infectious diseases, occur like in the brain and nervous tissue. So. All right, what is a reservoir? Yeah, place where you can find organism, okay, pathogen. And if you're a living reservoir, what do we call you?
Carrier.
A carrier, good. What is an asymptomatic carrier? Someone who has pathogen but no symptoms, right? So they can still transmit it, but they don't know usually because they are asymptomatic. What about an incubation carrier?
Is that somebody that has like some lesser signs and symptoms of the disease?
Right, because they just got infected, right? So they don't have many signs and symptoms because it's still early, but they still have the organism there, they can still transmit it. What about a convalescent carrier? Yep, you're recovering from disease, so you feel better, you go outside, you start talking to people, whatever, and then you spread the disease still. Okay, you still have enough of the organism present. What about a chronic carrier?
Always have it.
Oh, what's that? Yep, you always have it. So you were infected at one point, you probably had disease, but you're completely recovered now. But there's still low levels of it present, and you can still transmit it. And then passive carrier?
It'd be like somebody like in a hospital that is working with somebody with C. diff or whatever, and then doesn't wash their hands and those infected.
Right, so you don't actually get infected, but you still harbor it, and you can still transmit it, right? So getting stuff in your hands and then touching things and so forth. What about an animal reservoir? What do we call them? A vector, right? You can have a biological or a mechanical vector. What's a biological vector? Right, so they're actively infected. Okay, the vector, the animal gets infected, they can then transmit it to another individual. What about a mechanical vector? Passively, right? So they don't actually get infected, instead they pick the organism up and then they can transmit it to and from. What about a zoonosis?
They cause more serious diseases.
They cause more serious diseases, and where do they come from? Animals. So it's an animal disease that has gone from animal to a human. Non-living reservoirs, those are called non-living reservoirs. You guys are still awake, aren't you? Now, these guys can be communicable or non-communicable. Okay? Communicable means spread from one person to the next. Non-communicable essentially means it's intrinsic, right? So it was internal, and then it became pathogenic later on, for whatever reason. So for communicable diseases, you can have direct contact, spread, and indirect, okay? Direct requires close interaction, okay? So physical contact or being within about a foot of the person. Indirect, on the other hand, requires some sort of inanimate object or a non-living reservoir that the pathogen can hang out on for a period of time before it is then transmitted to another individual. What is a nosocomial infection?
Acquired in
Yep, you acquire the disease, the infection, in a hospital. What is the etiological agent of a disease?
Positive agent.
The organism that causes the disease, right? And again, this can be kind of hard to figure out, at least early on in an epidemic. And we do this by following Koch's method, right? So So isolation of the organism, reinfection into a healthy individual or animal, and then re-isolation, compare and contrast. Then lastly, what is epidemiology? So epidemiology is the study of disease and how it is spread. Okay? This is important so we can potentially stop the spread of disease. And then a couple definitions: epidemic, pandemic, endemic, and sporadic. Epidemic is a sudden acute outbreak, so a rapid outbreak of a disease in a geographical region. Pandemic is a sudden acute outbreak of a disease, but globally. Endemic is a low-level or chronic occurrence of a disease within a geographical region. And sporadic is a random outbreak of a disease. And then we can use this information to come up with the morbidity and mortality. What is morbidity?
When you have more than one.
How many people get sick? Right. So how many people get sick from that disease? That's the morbidity. What about the mortality?
How many people die?
How many people die? Good. All right. Dreaded immune system. So, chapter 14 deals with the innate immune system. The innate immune system are the first to respond. Remember, they don't get better over time. So they respond pretty much the same way every time they come into contact with the same pathogen. So innate, non-specific, but they're the first to respond. So without these guys, it would take weeks before you actually started to respond to pathogen. That would be too long and you would probably die. So they're very important in the fact that they respond quickly. Now, before you get infected, one of the easiest ways to prevent infection is to block it. Okay? You can use a physical barrier or a chemical barrier. Physical barriers just physically block the organism from gaining entry, or chemical barriers are chemicals that are released by the body to destroy microorganisms, preventing them from gaining entry. So now let's actually go into the immune system. Okay? So immunology, remember, this is the study of the cells that survey the body, recognize foreign material, and then destroy it. Okay? So survey, recognition, destroy. And they do this based on self and not self. So if it's self, it's allowed to live. If it's not self, it is destroyed. And remember, these are just a whole bunch of cells that are found all over the body, but primarily you're going to find them in the bloodstream and the lymphatic system. Okay, those are the two main areas you see. So what is the lymphatic system? It's a series of vessels and nodes that run parallel to the cardiovascular system. Its main purpose, at least physiologically, is to collect excess fluid, but it acts as a nice highway for immune cells. And remember that we break up where cells are found, or the tissues associated with cells, into two categories: primary and secondary. Primary lymphoid organs are areas of the body where cells are mature, okay, or allowed to grow. Where secondary lymphoid organs are areas of the body where immune cells are found circulating around the body, waiting for potential infection. Okay? Primary organs of thymus and bone marrow, and secondary organs of spleen and the lymph nodes, the lymphatic system. And lymph nodes are just openings of lymph vessels where why do I keep saying that? Where immune cells can congregate and communicate with each other. Thymus, which we talked a little bit about in chapter 15, and we'll talk a little bit more about later, is essentially an organ that is used for the maturation of T cells. Does anyone remember what maturation means? Maturing, right? Good. But from a T cell's perspective, what does that mean? So remember, when you make T cells, you're going to make cells that respond to self as well. So maturation is where you destroy those. So you're getting rid of all the self responders. Otherwise, they will cause autoimmune diseases. Okay, what is hematopoiesis? Production of blood cells, right? Using stem cells in what part of the body? Bone marrow, right. And so you can make red blood cells, white blood cells, or platelets. Which ones are the cells for the immune system? White blood cells. Now within the white blood cells, there's two groups, granulocytes and agranulocytes. What's the difference between those two groups?
Granulocytes have granules.
Right, you can see dots in them, little granules. Within the granulocytes, you have neutrophils, basophils, and eosinophils. What is a neutrophil?
It's like the garbage truck.
The garbage trucks, right? They're phagocytes. They're the most abundant immune cell. Their job is to gobble up foreign material and then die. Okay. What about basophils? What do basophils do? Allergic responses and then help clear large parasites. What about eosinophils? They're similar to basophils, right? They can cause allergic responses, and they can also get rid of large parasites. Then onto the agranulocytes. What are T cells? You can write that on the exam. Let's see how well you do. Yep, that's where they mature. So T cells develop cell-mediated immunity. Remember, that means they can help the immune response, but they can also kill cells that are abnormal or virally infected. T cells help the immune system, but also can kill cells that are weird. What about B cells? What do B cells do?
Make antibodies.
They make antibodies. They're also known as humoral immunity. And then monocytes, what do? They phagocytose, but what else can they do once they do that? Yeah. Right, because they can present the antigens to T cells. Because T cells cannot see antigen on their own. What do we call monocytes that go into tissues? On the next slide, if you really don't know. Macrophages, good. You guys are faking. I'm the sick one. I should be the one faking.
So you're making us all sick.
Hey, not my problem. Inflammation. What is inflammation?
Inflammation is the start of reaction.
So it's the hallmark of the immune system, right? And it's basically edema, or buildup of fluid in a tissue, right? And this can cause redness, warm, swelling, pain, all that yada yada yada. Why do we have inflammation? It recruits immune cells to the area. Okay, remember, immune cells are found scattered throughout the body. So if you didn't have inflammation, they would not know where to go. Okay? And inflammation causes vasodilation, or opening up of blood vessels. This is what creates that edema or buildup of fluid, but it also allows immune cells to get into the area. What are the first immune cells to come into the area during inflammation? Neutrophils, right? The garbage trucks. They come in, they gobble up the whatever they see, and then they die. And if they don't get it under control, who then comes to the rescue? Sometimes, right? But mostly macrophages and B cells, okay? I'm bringing this up now because you're going to see it again when we talk about chapter 13. And if you can't tell me it when I ask you then you're not listening. And what brings on inflammation? What starts inflammation? Tissue destruction, right? And that tissue destruction brings out cytokines and chemokines. These are proteins, chemicals, that start inflammation and also attract immune cells to the area. And they attract immune cells to the area via chemotaxis or following a chemical signal. Okay? So, having this inflammation and edema allows for the attraction of immune cells to the area, but it also dilutes out any toxins that are present, allows for clotting, and gives more volume for phagocytosis to occur. And again, if the neutrophils don't get it under control, then the macrophages, B cells, and T cells can come in and start to fight off the infection even more. All right, phagocytosis. What is phagocytosis? Engulfing the foreign thing, right? And what cells can phagocytose? Monocytes, neutrophils, and macrophages, right? And sometimes B cells. So, what starts phagocytosis? How does the cell know to phagocytose or gobble up? Chemotaxis, based on, what is it looking for?
Chemical signal.
A chemical signal. Well, what's that chemical signal called? A PAMS, right? Pathogen-associated molecular pattern. So it's looking for something on the surface of the organism that looks bad. Okay? Once it finds that, it follows it, chemotaxis, then binds to it, swallows it up, fuses with the lysosome, degrades it, and poops it out. Okay? Fever. What is fever? These easy ones, you guys should just be able to spout out in 2 seconds.
High body temperature brought on by inflammation.
High body temperature brought on by systemic inflammation, right? Why have it? What's the benefit of having fever? Yeah, it can slow the organism's growth, right? Because they're highly dependent on temperature. What else?
Tells your muscles to be more metabolically active.
Right, so it tells all the cells in your body to be more metabolically active, including immune cells, so they work better and faster. What else? The one that always people always forget, unless you're reading it.
Peripheral arterial secretion loss through
That's how you. So that's how you can decrease fever. But what is the other benefit of fever? It's on the next slide.
It keeps the nutrition.
Right. It keeps red blood cells occupied. Okay, because they're busy, and this reduces the likelihood they'll be eaten by bacteria. Interferon. What is interferon? Right, it's a protein that is released by cells that are infected by a virus or intracellular parasite, and it acts as a warning system, okay, telling neighboring cells to prepare themselves. What about complement? What is complement? Right. Blood proteins produced by the liver that poke holes in bad cells. Okay? And you can have either classical complement or alternative complement. How does alternative complement work? What do you need? This one's not written in the slides because we just talked about it. Classical requires an antibody. Okay? So when an antibody is bound to the cell, complement then forms and pokes a hole. What about alternative complement?
You don't need an antibody.
What's that? You don't need an antibody, right? And it's based on the cell not removing it, okay? So if it lands on one of our cells, we remove it, we don't get a whole poked in it. But if it's a bacteria, it lands on it, cell doesn't remove it, and a whole then gets poked into. All right, so now let's go to 15, and this will summarize the entire immune response. Get this way so you can see the slide numbers. So, 15 deals with the third line of defense, also known as the adaptive immune response, or acquired immune response. This is extremely specific, right, down to single species level, and it gives you immunological memory, or the inability to get infected with the same pathogen again. So let's go to this slide. This is a great summary slide of pretty much this entire chapter, okay? Now, we've got two parts to this, the orange part and the blue part. What is the orange part telling us? Right, so B cells replicating and making them, right? So the development of B and T cells. Where do you make B and T cells? So that's where they mature, but where are they made or produced? Where's hematopoiesis? Bone marrow, okay? So all blood cells are made in the bone marrow. Both B cells and T cells are made in the bone marrow, right? And when they're made, what do they do to themselves so that they respond to a unique antigen? Not yet. They what? No, not yet. So before that step, how do they determine their antigen? Remember, it's a random shuffling of genes. So they randomly pick a couple genes that they have, push them together, and now that's their antigen. Completely random process. Now we can go into the bone marrow and thymus. So this is where we go through a process called maturation.
Is that why you said, they go around looking for a certain strain? Not strain, I'm using the wrong word.
Sorry.
A certain
Antigen?
antigen on a T cell?
Right, so that's what will happen after they go through this process.
Okay.
So in the beginning, during development, what you do is these T and B cells change their genetic material, so each one of them responds to a unique antigen. Some of those antigens will be your own. Other antigens will be everything else outside. And what you need to do during maturation is get rid of the guys that respond to your own antigens. If you don't do that, they will kill your cells and cause autoimmune diseases. So maturation, thymus for T cells, the bone marrow for B cells, is where you are eliminating the cells that respond to self, or your own antigens. Again, if you do not do this, these cells will be released into the body and cause autoimmune illnesses. So let's just talk about this one more time. You guys are all looking at me like I'm crazy. So, B and T cells are made in the bone marrow, okay? During this process, they randomly select genes to make an antigen they respond to. Some of these antigens will be your own, and some of them will be to outside things. We want to get rid of the ones that respond to our own antigens, otherwise they will kill us, okay? So to do that, you go through maturation. T cells go to the thymus, B cells stay in the bone marrow. During maturation, you show all of your antigens to these cells, they respond, you kill them. So what you're left with are just B and T cells that respond to everything else. Okay? Can I make it a little clearer? Okay. Yeah. All right. Now the blue part. What is the blue part?
Infection.
Infection. This is what happens after you get infected, right? So let's take it a step back and bring in chapter 14. Okay? So let's say I got a needle, jab it into my arm. What's the first thing that happens? Inflammation. Brought on by Who started the inflammation? Tissue destruction, right? So tissue destruction induces inflammation. This recruits who? The area. Neutrophils, right? They gobble up all the stuff, but if they can't get it under control, then who comes in? B cells and macrophages. Now that brings us to here, okay? B cells and macrophages go to the area, and they look for pathogen or antigen. And if they see it, what do? Destroy it by phagocytosis, right? They gobble it up. Now when they gobble it up, they break it down, and then what do they do with it? Do they just poop it out?
No, I think that's one.
They process it and present it, right, on the surface. Now for B cells and macrophages, what molecule do they present it in? Begins with an M, ends in HC.
MHC.
MHC. Okay? And remember, there's two types of MHC, class 1 and class 2. Anyone remember which one is used by macrophages and B cells?
Class 2.
Class 2. Good. You had a 50-50 shot and you got it.
I
Okay?
remember you saying that. Sort of.
So, it's processed, presented on MHC class 2. Why do they do this? Who needs to see antigen in MHC class 2? T cells, right. What type of T cell? CD4, good. So CD4, MHC class 2. CD8, MHC class 1. Okay. So they phagocytose, process, present an MHC class 2, and then go into the lymphatic system to show it to T cells, right? In hopes that they find a T cell that recognizes it. When they do, what happens? No. They dump crap on each other and they get all happy and excited. Okay? The T cell then goes, makes many copies of himself, most of which will go off and help fight the infection. Small percentage of them will become memory cells that give you immunological memory for the time you get infected with this again. Okay? So there are the T cells. Now let's go to the B cells. So B cells also came to the area, right, where the infection is. If they recognize that pathogen, they can phagocytose it. But can they do anything after that? No. They need the T cell to fully activate them before they can continue on. So they then process, present it to T cells, and if they find the T cell that activates them, they become happy, make millions of copies of themselves, most of which become plasma cells, which do what? Plasma cells said what? They make antibodies. Right? Plasma cells make antibodies. And then a small percentage of them become memory B cells, which give you immunological memory. Right. Memory cells give you memory. Surprise, surprise. Okay, so let's go through this part of it, right, in a couple brief sentences, right? So, jab myself with the needle, causes inflammation, recruits neutrophils, and they gobble up the bacteria. If they can't get it under control, then macrophages and B cells come in, phagocytose, process, and present on MHC class 2. They then show this to T cells until they find the one that recognizes it. This T cell then becomes activated, makes many copies of themselves, most of which will help fight the infection, and some will then sit there and wait for another secondary infection or another immune response. The B cells, on the other hand, are doing the same thing. They're presenting it to T cells in hopes that they find a T cell that recognizes that antigen. They do. They become activated, make millions of copies of themselves, most of which become plasma cells that make antibodies, and a small fraction become memory B cells that can lie in wait for a second infection. So, let's see, what did we forget to talk about? So So this is just an example of that maturation step I told you about. You're getting rid of cells that respond to self, so they do not cause autoimmune disorders. Talked about all this already. Let's just skip through it. All right, so what are antibodies? They're Y-shaped proteins that stick to stuff. Okay? They stick to antigen. Why do they do this? These 6 reasons. This is an important slide. Slide 46. This shows why antibodies are important in fighting infection. They tag cells, which piss the bacteria off. Makes it hard for them to grow. This also makes them tastier to phagocytes, so they can gobble them up and destroy them quicker. They can agglutinate or stick multiple organisms together, again, pissing them off. They can fix complement. What type of complement? Does anyone remember? Classical, right? You need an antibody for classical. They can neutralize viruses, blocking the virus so it can't get into the body, or into the cell, excuse me. Or they can precipitate or bring out small molecules out of solution so they no longer are toxic to cells. And remember, there's 5 types of antibodies: IgG, IgA, IgM, IgD, and IgE. Where do you find IgG?
Blood.
Blood. It's the most abundant in the blood, right? It's kind of the most ubiquitous. It's the best at doing pretty much everything. Okay? What about IgA? Where do you find IgA?
Mucous membranes.
Mucous membranes. So what do you think its job is? Prevent infections at the mucous membranes. What about IgM? Where do you find IgM? The blood. It's the first antibody to be produced, and it's 5 IgM stuck together, which means it's really good at complement fixation and agglutination. What about IgD? Where do you find IgD? Yep, it's a B cell receptor. Okay. What about IgE? Basophil mast cells, right? So it's a mast cell receptor. So you probably shouldn't see much of that in the blood. Okay, good. 52. So this is a nice summary slide describing immunological memory. Okay. So in this slide, you have your initial infection, which is the first peak, and then your second infection, okay, which is the second peak. First peak, you get infected, right? And there's this long period where you're not making any antibodies to that infectious agent. Why? Yeah, you have to go through that entire process we just talked about, from start to finish. And that takes weeks, sometimes, okay? So initially, there were very little antibodies, okay? Then you start to make IgM and then IgG, okay, once everything gets rolling. And then once you clear the infection, those plasma cells die, and you're left with, again, very little antibody, okay? Now, what got produced during this primary infection other than antibodies? Memory cells, memory B and T cells. Those cells circulate around the body, dormant state, doing nothing, waiting for you to get infected for a second time. Now we get infected for the second time. What happens? Much faster because the memory cells respond right away, right? So they see it, they go, oh crap, I know this guy, I'm going to go crazy right away, okay? And so immediately, well, relatively quickly I should say, you start making tons of antibody. IgG first and then IgM, and you make more antibody the second time around than the first time around. So the combination of more antibody and very rapid response, the production of antibody, results in clearance of the infection before you get sick. Okay? So this is why we can have immunological memory. All right, so T helper cells, these are CD4 cells, and as their name implies, they help the immune system, primarily B cells, production of antibodies. Cytotoxic T cells, also known as CD8 T cells, these are cells that destroy any cells that are abnormal, right? So cancer or virally infected cells. Okay, so real quickly, a little bit about vaccination, and then we'll be done, okay? So vaccination can occur both in active and passive form. What do I mean by active?
Actively produce memory cells.
Right, so you actively produce memory cells, you go through all the stuff we just talked about, right? You make your own memory cells and then you get long-term protection. The benefit of this, long-term protection. The drawback to this, it's slow. What about passive immunization for vaccination?
Short term.
Short term, right? So it's very rapid, however, it doesn't last for very long. And again, this is where we take antibodies from another source and use those instead of producing our own. And both of these can be either natural or artificial. Natural active immunity is where you get sick, right? So you get a cold or a flu and then you develop your own immune response to it, and now you're protected at least somewhat. And artificial active immunity is a vaccination, right? Getting a shot in the arm and then developing your own immune response. Natural passive immunity, this is where you pick up antibodies from your mom through colostrum or breast milk or from the placenta. And artificial passive immunity is where, again, we inject preformed antibodies into the body. Okay. Now, as far as vaccines are concerned, there's two main groups: whole cell or a subunit or genetically engineered vaccines. Okay. By far the most common are the whole cell, and in this there's two flavors: live and kill. What's the benefit of using a live vaccine? So yeah, there'll be antigens. You get a stronger immune response, okay? Because the organism is alive. So you get a real immune response. It's also at the site of infection. What's the drawback to using a live vaccine? You can get sick, right? Good. What about a killed vaccine? It's not pathogenic, so you're not going to get sick. But the drawback? Your immunity isn't as strong, right? And it's often in the place where you don't get the natural infection, right? In your arm or in your butt or something like that. What about a subunit vaccine? What is this?
Take the antigen instead of the entire cell.
Right, so just the antigens, not the whole thing. Do these work well? They're okay, right? So we typically need to give a lot of these before you develop true immunity. Don't worry about all the genetically engineered crap. And remember, vaccines also have other additives, primarily adjuvants. What's an adjuvant? No? So Thermacell is a preservative, okay? Adjuvants in general are just compounds that induce an immune response. So it gives you a better immune response, okay? But there are preservatives in there, right? And those prevent other organisms from growing within the vaccine itself. And these can often cause some small, minor side effects. And last but not least, what is herd immunity?
Mass immunity.
So it's vaccinating the majority to protect the minority, right? So if you vaccinate the majority of the people, it slows the pathogen down because there's not enough people for it to spread to and then cause disease widespread, okay? And so this results in protecting the very young who can't be vaccinated or the very old who have a weakened immune response. Okay, that is it for this. Any questions on anything? Cool. This is everyone's favorite.